This is the figure that I want. I think this will help us illustrate LTP in the hippocampus. Remember the hippocampus, a target because it's an area where we know we see plasticity, right? Plasticity would be required in learning or memory, and we know that the hippocampus is part of that circuit. So let's set up this experiment a little bit, okay? The way that they first demonstrated LTP in the system. Well, so the two neurons in the circuit that we're going to talk about are in different regions of the hippocampus, right? So one of the neurons that we're going to mention are those that are in the group N, the CA3. Yes, this is the figure that I want. I think this will help us as we discuss LTP. And remember, we're looking in the hippocampus, right? The hippocampus we know to be involved in learning and memory. And learning and memory, you would sure think, would require changes in synaptic strength, right? That's what's going to encode our memories. So let's set up this experiment, and let's just remind you of the circuit here first, right? There's a really commonly well-characterized circuit between CA3 axons, right? So these neurons have their cell bodies in CA3, but those CA3 axons project to CA1 dendrites, okay? So again, the CA1 bodies are there, but those dendrites might not be exactly next to the cell bodies, right? So you have CA3 axons projecting to CA1 dendrites. This is the Schaefer collateral pathway, CA3 to CA1. And what you can do is you can stimulate CA3 and measure the response to that stimulation, measure in CA1. Okay, so this is just generally how this is performed. So let's look at the experiment. Okay, what they're going to do is they're going to have a control cell, right? And they're going to have an experimental cell from CA3. Okay, so CA3 will have two axons. One control, and one experimental that they're going to investigate in this study. So one experimental. And in either case, the first thing that they do is they're going to stimulate and record a baseline response. They're going to stimulate the CA3 axon. So stimulate the control, stimulate the experimental, and again, measure the effect postsynaptically. So stimulate, then measure the EPSP, right? So this EPSP is being measured in the CA1 neuron. And the same is going to be true for the experimental. Stimulate and then measure EPSP of the CA1, or in the CA1 neuron. And so you can see that they do that here in pathway 1, right? This is our experimental group. So we see that the cell is resting at about -60. They stimulate CA3 and they measure this response in CA1, about a 4 millivolt EPSP. Okay. And when they do this in their control, it looks very much the same. Here's the stimulus right here, and nothing's happening, cells at baseline, and now we notice this EPSP. Then the experimental neuron gets tetanized. Okay, so it's going to get high frequency stimulation to its axon, whereas this one, the control, doesn't get any of that tetanus. So no HFS, no high frequency stimulation. Okay, and after that, right after they provide the high frequency stimulation, they stimulate and measure again. And so they're going to stimulate and measure in both. And in the control, when they provide this stimulus, their response looks exactly like it did the first time. about a 5 millivolt EPSP measured in CA3. Excuse me, in CA1. Jeez. All right. Well, what about in the experimental? Well, now after the tetanus, right, here's the stimulus that they give right here. You can see a substantial increase in the EPSP, right? So instead of maybe a 4 millivolt EPSP here, right, if we go across, they're getting almost a 10 millivolt EPSP. This is the beginnings of what we'll come to call long-term potentiation. Now, of course, how long does this last? That's the next question. But we see this enhancement, right? We see this increase in signal strength here after tetanizing one of these neurons in CA3 that has an input to the CA1 dendrite. So we want to know, is this long-term? How long does this last? Length of potentiation, effect. Length is a bad word. I want duration. I want to know how long it lasts. Okay. So panel C is from the same group of experiments as I showed before. So again, we're measuring the EPSP, so this has to be measured in CA1. And what we're doing is we're giving control pulses to CA3, right? So we have CA3 pulses, right, little lightning bolts, right, to CA3, and we're measuring the response in CA1, okay? And so the purple group, these purple dots, indicate individual CA1 responses. And they're doing this as a percent of control, okay? So what we see is that when we give a zap to CA3, the CA1 response might be 5 millivolts, right? And then the next one might be 5 millivolts. And then the next one might be 5 millivolts. And then maybe one's 5.1 millivolts or 4.8 millivolts, okay? So we're calling whatever our initial response is 100%. Then you can see that they give one of these connections, one of these CA3 axons, high frequency stimulation. And now, when they give an individual stimulus, just like we saw before, we see potentiation. We see a stronger response in CA1, right? This is where we're measuring. So, for example, if the CA1 response pre-tetanus was, what do we want to say? I don't know, maybe 5 millivolts, right? That's how much depolarization occurred. Right away post-tetanus, we max out at about 300% that. Increases 300%. Right, so that's like 3 times the effect. Now we'd likely see that same stimulus given, and it wouldn't give us a response of 5 millivolts seen in CA1, right? The depolarization seen in CA1 would be 15 millivolts, okay? And now let's look at the duration. If we're going out an hour, after 1 hour, right? So we're talking 1 hour post-tetanus, right? Post high frequency stimulation. There's still a 200% increase compared to controls, right? So we're still at twice as much of an effect. That is, the EPSP is still 2 times the effect that we would see under control conditions. Okay, and you can see here that in yellow, this other pathway that doesn't get any high frequency stimulation, this never really changes. Okay, all right, so we know it lasts at least an hour. Well, take a look at the bottom panel. Now, our time scale is in days. Okay, So again, they're going to do the same experiment where they're going to stimulate CA3 and then measure the responses in CA1, okay, as a percent of control. And so you can see on that very first day, right, after HFC, they're still getting a response that's greater than before the HFC. And this effect lasts for a year or longer. So even after 1 year, the response in CA1 to CA3 stimulation after high-frequency stimulation is still potentiated. Now, it's not potentiated by a factor of 100, it's not a 100% increase or a 200% increase. No, it's not. It's modest, right? So it's about a 110%, so it's about a 10% increase from before. So instead of seeing a 5 millivolt EPSP, maybe they get that plus a little bit, maybe it's 5.5 millivolts, but it still is there after a year. So I think that's pretty incredible. So let's talk a little bit more about this. Let's talk about 3 unique properties of long-term potentiation. Okay. The first that I would mention is that it requires activity that's strong pre and post-synaptically. So pre and post-synaptic activities. So I think we've seen how presynaptic activity can lead to LTP, but what do I mean postsynaptic activity? Well, let's look at the experiment to the right, which is similar, right? It's the same CA3 to CA1 circuit that they're going to measure. And now that the stimulus that they provide to CA3 is going to be relatively slow, okay? So there won't be any high-frequency stimulation, only a few stimuli to CA3. So we're going to have relatively little stimuli of CA3. And as you would expect, this fails to induce sorry, I lost my train of thought mid-sentence. The dog is here with me at Lakeland and just was going crazy. So I had to go check and see what it was. It was just another dog. Okay, so what they're going to do is provide individual stimuli, right, and then measure the response in CA1. And as you would expect, little stimuli of CA3 equals no long-term potentiation. And so you could induce long-term potentiation by giving this CA3 axon tetanus, but instead, right, in this case, what they're going to do is provide brief polarization of CA1. So now they're going to stimulate CA1. So they're still going to give these individual depolarizing pulses here, but paired with EPSPs that they can also generate from this recording electrode here. Okay? And doing that triggers LTP, right? Again, we're looking long term, so we have to look minutes out. And so you can see that it's the combination of both that are going to be necessary to have an effect. So what can we say here? So when CA1 stimulation is paired with the depolarizing pulses, to the CA3 axon. Right, this induces LTP. We'll talk about mechanisms of this in a second, but I want to point out here that we get both pre- and postsynaptic activity, right? Now, activity isn't necessarily depolarization, but something changing. Now, in this case, it was depolarization, but we'll see what changes postsynaptically when we talk about these mechanisms, okay? So what I want to leave you with in terms of this figure is that we don't have to tetanize the CA3 axon to induce LTP. A little bit of stimulus, not even a little bit, but some depolarization of CA1 can trigger LTP. This is known as Hebbian plasticity, based on the person who first described it. And the phrase that is associated with Hebbian plasticity is neurons that fire together, wire together, right? And we'll see how the wiring changes in a minute, but certainly the firing patterns of these neurons are linked because when they fire closely in time, we can see this LTP develop, okay?